Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So we were discussing cellular signaling, in which we have covered the basics part, like what is cell signaling and how the cells they are able to communicate with each other. So we learned about the basic schematic, the basic way of a signaling cascade. What are the basic elements that are included in the cell to be able to receive a signal and to be able to respond accordingly. Again, we also learned about molecular switches, how they play a very important role in the signaling cascades. Today, we'll be learning about GPCRs, the G protein coupled receptors that are present on the cell surface. So, in the last class, we learned about how the signaling cascade goes on. Whenever any extracellular signal molecule comes, it goes and binds to the receptor. So, for a cell to receive any kind of signal, it needs the receptors on its cell surface or intracellularly. So there are two types of receptors that can interact with the ligand depending upon whether they are present on the cell surface or whether they are present in the cytoplasm of cell. If they are present on the plasma membrane of the cell, they are referred as the cell surface receptors, and if they are present intracellularly, they are referred as the intracellular cell surface receptor, right? So we are going to discuss only about the cell surface receptors, which are of different types, such as GPCRs, the enzyme coupled receptors, and inotropic receptors. The inotropic receptors, which are also known as ion channel coupled receptors. We have discussed about them in our neurobiology series, so we are not going to discuss that here, but we'll discuss about GPCRs and enzyme coupled receptors. So in today's class, we are going to discuss about G protein coupled receptors. So in yesterday's class, we discussed about the molecular switches, in which we discussed about the G protein as well as kinase and phosphatases. So we discussed how those protein molecules, they are able to get activated or inactivated, and they act as a switch on and off so that they can turn on the gene expression or they can turn off the gene expression, right? So we are going to today learn about the whole signaling pathway of the G protein coupled receptor, how the certain gene expression is activated with the help of those receptors, right? So this is a very large family of receptors, and due to those G protein coupled receptors, you are able to sense, you are able to have vision, you are able to sense the smell. G protein coupled receptor, you have your sight, you have your smell sensation. Okay, so the rhodopsin molecules that you have been learning about rhodopsin from your high school level. So they are the examples of G protein coupled receptors. So this is a very large family of the receptor proteins. So despite their chemical and functional diversity, the basic structure that G protein coupled receptor GPCRs that they have, that is same across the biological systems. So the basic structure of GPCR is that it has a receptor, it has a G protein, and it has an enzyme. Okay, the enzyme which can be activated by the activated G protein. Okay, so we are going to discuss about that in detail, and we are going to see that how the signaling goes on. So this is a schematic that I've taken from Bruce Alberts. Here you can clearly see that this are the three subunits which are in the inactivated form. So we have a receptor, okay, obviously, which is going to interact with ligand. The signal molecule is going to go and bind to the this receptor, and this receptor is coupled with a G protein, and that is why it is known as G protein coupled receptors, right? So the receptor which is being coupled with G protein, it has certain characteristics, and it is same across all of the different types of cell. The characteristic is that it is a 7-pass transmembrane protein. Okay, means it passes the plasma membrane 7 times, right? So this inactivated receptor, it is a 7 pass, right? 1, 2, 3, 4, 5, 6, and 7. So it passes the plasma membrane 7 times. So this receptor is coupled with a G protein, and that is how a GPCR is formed. So here you can clearly see that this G protein is heterotrimeric, as we discussed in last class. It has 3 subunits: alpha, beta, and gamma subunit. The alpha subunit has 2 domains. Okay, the RAS domain, this is known as RAS domain. Okay, and this is AH domain. Okay, the RAS domain, this domain, it binds with the receptor. Okay, so this binds with the receptor. Here you can see that RAS domain helps the binding of G protein with the receptor, while the AH domain here, this AH domain, it binds to the GTP or the GDP. Okay, so this inactivated G protein means this is bound to the GDP. This is bound to the GDP. So we learned in the last class how this is able to switch on. So whenever this GDP is being replaced by a GTP with the help of guanine exchange factor, the enzyme GEF, when GEF works on this inactive G protein, the GDP which was situated in this AH domain, that will be replaced with the GTP. And when it is replaced with the GTP, this G protein, it gets activated and it splits into two parts, the alpha subunit and the beta gamma subunit together. So the alpha subunit, which is bound with GTP, it is going to activate the inactive enzyme, which is going to do the downstream signaling. Okay, so this G protein, the G alpha subunit, is going to activate certain enzyme, and the beta and gamma subunit, they also show some downstream effects. Okay, so we are going to discuss about this signaling pathway in detail. So depending on which type of G protein this receptor gets coupled with, there are four types of different signaling. Okay, so there are four different, like four main categories of G proteins. Okay, the GS, which is also called as stimulatory G protein, the GI protein, G12-13 protein, and GQ protein. Okay, in today's lecture we are going to focus about the signaling of stimulatory G protein only, and in next lecture we'll discuss about the signaling pathways of those three G proteins. Okay, so though these are different, okay, these G proteins, even though they have same, same structure, like all of those three, they have alpha, beta and gamma subunit, but the way of functioning is very different. That is why they have been categorized into four main categories. Okay, so the GS protein is known as stimulatory G protein. When it gets coupled with with a receptor, okay, when this GS protein gets coupled with the with a 7-pass transmembrane protein, it stimulates an enzyme known as adenylyl cyclase. Okay, and that is why it is referred as stimulatory G protein, because it is going to activate the inactive adenylyl cyclase. So let's learn about how this overall process goes on in the cell. Okay, let me draw it for you. So this is the plasma membrane of cell. Okay, and this is a 7-pass transmembrane protein, the receptor. Okay, so this is coupled with the G protein. The G protein has three subunits: alpha, beta, and gamma. So this, the G protein which it is being coupled with, is the GS protein, and this is the receptor, 7-pass transmembrane receptor, and this is the plasma membrane. Okay, so let's say that some extracellular signal molecule arrives, and it interacts with the transmembrane domain. So when it interacts with the receptor, this inactive this inactive GS protein gets activated. So what happens is that the GDP, which was bound, the GDP, okay, GDP, which was bound with the alpha subunit, it gets replaced. Okay, GDP leaves, it gets replaced with the GTP. Okay, so once the GTP binds to this alpha subunit, this G protein splits into the two parts: the alpha subunit, which is bound with GTP, plus the beta and gamma subunit. Right, so this alpha this alpha bound GTP subunit, it separates from the receptor. Okay, it separates from the 7-pass transmembrane receptors, and now this alpha bound GTP, it is going to activate enzyme called adenylyl cyclase. Okay, so this adenylyl cyclase, this enzyme, this catalyzes a reaction, the conversion of ATP to the cyclic AMP. This adenylyl cyclase, this catalyzes the conversion of ATP, adenosine triphosphate, into the cyclic AMP, and the cyclic AMP, which is a secondary messenger. So this is how the primary the primary message, which is this extracellular molecule, it results into the formation of secondary messenger, right? So I hope you now it is you now can have a clear picture about the signaling pathways, how a primary messenger results into the formation of secondary messenger with the help of a molecular switch. And here the molecular switch is the GTP binding protein, or the G protein. And the which type of the G protein it is? It is the GS protein, the stimulatory G protein, right? And this is how the secondary messenger is formed. So now the secondary messenger has, has a lot of jobs to do. So this is going to activate certain genes. So the genes that are going to be activated by the CAMP, they are referred as CAMP. So the gene region, the gene element that is going to be responding to CAMP are known as CAMP response element. Okay, so this is so whenever we say element in the context of DNA, this is the part of, this is the group of different types of different genes. They come together, and they they are stimulated due to certain factors, such as here they are stimulated due to CMP, or they are stimulated in under certain condition. So they are the group of genes which are which are stimulated due to certain condition, or which result into same effect, and that is why they are referred as elements. So they are a group they are a set of genes that are present on the on the chromosome, on the DNA, right, on the in our genetic material. So that is why they are referred as element. So the set of the genes, or set of the set of the genes which are present in our DNA, which gets activated or stimulated due to these the CAMP, okay, this secondary messenger, they are referred as CAMP response element, which is abbreviated as CRE. Okay, CRE. It is referred as CRE, the CAMP response element. Okay, so let's now learn how the CAMP is able to activate this CRE, the CAMP response element. So what happens is that when the whenever the secondary is whenever the primary messenger molecule, the extracellular signal arise, it is going to result into the splitting of the G protein, and now this will result into the activation of adenylyl cyclase, and thus the CAMP levels will increase inside the cell. Okay, so once the CAMP levels they increase inside cell, it activates a protein kinase known as pKa. So this is protein kinase A. Okay, so this secondary messenger, the CAMP levels, the high CAMP levels results into the activation of this enzyme, protein kinase A. So it is a kinase, means it is going to phosphorylate certain enzyme. So let's first now learn about the structure of pKa. So let's say that this is a pKa molecule. So this pKa molecule has two regulatory domains. Okay, so these are the regulatory domains. Okay, and it has two catalytic domains. These are the catalytic domains. So as it is obvious from the name, regulatory domain means it is going to regulate the function of the of this kinase, of this enzyme, right? And whatever enzymatic activity is going to be done, that will be done by the catalytic domain. So for activating this catalytic domain, you need to stimulate or activate the regulatory domains, and the activation of regulatory domain is done by CAMP. So what happens is that the CAMP molecule, okay, the four CMP molecule are required so that the PK is activated. Why? Because what happens is that whenever the CAMP level increases in the cell, so the increased CAMP, those molecules, this CAMP molecules, they go and bind to the regulatory domains of protein kinase A to two molecules of CAMP. So this is the CAMP molecules. Okay, these are CAMP molecules. So two molecules bind with one regulatory domain, and other two molecules bind with the other regulatory domain. So you need four CAMP molecules so that the regulatory domain of the protein kinase A is activated. So the binding of this, binding of the CAMP molecule with the regulatory domain, it triggers the splitting of this protein kinase A. So what happens is that this regulatory domains, they stay back inside the cell. So this is the regulatory domain. Okay, they stay back inside the cytoplasm. Well, those catalytic domains, these are the catalytic domains of protein kinase A, they go inside the nucleus. Okay, so they go and they translocate inside the nucleus. Okay, regulatory domain stays back inside the cytoplasm, while the catalytic domain, they translocate towards nucleus. So once they translocate to the nucleus, so inside nucleus there's genetic material, right? So this is our genetic material, the DNA, and in this DNA, this particular region which I'm highlighting with yellow color, this region, this is known as CRE, the CAMP response element, because eventually it is the element, it is the part of our genetic material which is which is getting activated due to the CAMP. So what if the CAMP level increases, the CRE genes, they are going to get activated, right? So this CRE region is sitting inside the nucleus in our genetic material. So what happens is that this catalytic unit, so this is a catalytic unit of a kinase protein, right? And kinase, what does kinase do? This enzyme has one job. It has the job of phosphorylation. So it phosphorylates certain element, certain protein, not element, certain protein, right? So the protein it is going to phosphorylate is known as okay, it is known as the protein which is going to bind to this CRE element is known as CREB. Okay, so the protein which is being phosphorylated by this catalytic domain is CREB, CREB, which is known as CRE binding protein. Okay, the nomenclature is very simple and very straightforward. The protein which is going to bind to the CRE, the CAMP response element, it is known as CREB, the CRE binding protein, and this CREB is phosphorylated, and due to the phosphor. Okay, so what happens is that the CREB is there. The protein is being synthesized in the cell, so it is there, but it is unable to bind to the CRE element. So this is a transcription factor, right? So for the activation of this transcription factor, you need a co-activator, and that co-activator is known as CBP. CBP is CREB binding protein. Okay, again a very straightforward name. CREB binding protein is CBP. So you need a co-activator and you need phosphorylation event. So what happens is that the CREB protein, which is sitting inside our cell, this CREB protein is being phosphorylated, and due to phosphorylation of this CREB protein, it gets activated. Okay, and due to this phosphorylation, a co-activator known as CBP is able to bind to the CREB, and that is how this transcription factor CREB is able to activate it is able to activate the CRE, this CMP response element, and the downstream genes, they are being transcribed. So this this kinase catalytic domain, it phosphorylates CREB, CRE binding protein. So the phosphorylation of CREB results into the binding of CREB to CRE. So this is CREB, which is phosphorylated. So I'm drawing P. I'm representing P for the phosphorylation, and P represents phosphorylation. So this is the phosphorylated CREB protein, and due to the phosphorylation of crepe protein, now the co-activator of crepe protein known as CBP. Okay, so this is the catalytic domains which are, which are phosphorylating crepe protein, and due to this phosphorylation event, the activation of crepe is seen, and that activation is due to the co-activator known as CBP. Okay, the crepe binding protein. CBP means the crepe binding protein, right? And when this whole complex, this whole complex assembles inside the nucleus in our cell, then it triggers the transcription of the SCAMP response elements. So the downstream effects that are seen. So it usually transcribes certain genes which are regulated by CMP. So for example, it transcribes a hormone known as somatostatin. Okay, so we are not discussing about the effector proteins that are being formed due to this scenario. The point of focus was, how the signaling cascade goes on. Whenever a primary signal arrives, whenever a primary signal arrives, it is activating the receptor. It is activating whenever it is binding with the receptor. It is activating the G protein. The GDP is being replaced with the GTP, and this activation is again stimulating an enzyme which is producing a secondary messenger. And now this secondary messenger, due to the formation of this secondary messenger, again a protein will get activated, an enzyme will get activated, and that enzyme is going to bring out some catalytic changes, some metabolic changes, which is going to result into the transcription of the genes which are being controlled by this secondary messenger. That is why we call those genes, those somatostatin and whatever downstream genes that are being affected, that are being controlled by this element, as the CAMP response element, because they are being controlled by the CAMP, the cyclic AMP, and that is why we refer them as the CRE, the CAMP response elements, right? So this is how the secondary messenger has its effect downstream, and this is the basic signaling cascade that is seen by the GS protein. So the other type of G proteins are the GI proteins, which is inhibitory G protein, then G12, G30, G12-13 protein, and GQ protein. So today I conclude this lecture at this point. We just focused about the GS protein. Tomorrow we'll discuss about the other signaling. Today we discussed about the GS signaling. Tomorrow we will discuss about GI signaling, GQ signaling, G12 and 13 signaling. Okay, till then, see you. Bye bye.